is selected and put into a spare hive body in which the other frames contain foundation and drawn comb. Meanwhile, the other brood chamber has been examined for queen cells. The hive is now reassembled in the following way. The new brood chamber with the single frame of brood is placed on the original floorboard and the honey super on top. This is the queenless part of the colony, and all flying bees will soon return to it. A crown board separates both parts of the colony. The queen right brood chambers are put on top. All flying bees will return to the familiar original hive entrance over the next few hours. This weakens the queen-right nucleus to such an extent that its swarming fever is extinguished. After 7 to 9 days, the emergency queen cells, which have been raised on the single frame of open brood in the lower hive body, are destroyed. Another frame of open brood is introduced in its place. This keeps bees content and avoids losses through drifting. The two halves of the colony are reunited after a further 7 to 9 days. The swarming tendency is extinguished and the queen shows full laying activity. The hive is reassembled in the original way. Once again, any emergency cells found on the single comb of brood must be destroyed beforehand. This method of swarm control permits the continued evaluation of the queen and of her colony. Naturally, the control of honey production is the paramount consideration. It can be ascertained by frequent weighing of colonies during specific nectar flows. Any changes in the colony must of course be taken into consideration, such as additional supers. The use of clearer boards and bee escapes simplifies the removal of the honey crop. One day before supers are collected, the clearer boards are put in position. Bee escapes allow trapped workers to pass down into the brood chamber, but prevent their return to the supers. Supers can be collected the next day. The honey yield is established by weighing the supers. The origin of the queen and the purity of her matings is checked by testing her progeny morphometrically. In order to make quite certain that only the queen's own descendants are sampled, only freshly emerged bees and drones are taken off the frames. About 50 bees and 50 drones are required for a full assessment. Because the chitin of the young bees is still too soft, they are held in cages for a few more days before the samples are preserved for a later examination. Another aspect of performance testing is the selection of vital and disease-resistant stocks. Only they can produce optimum results. Among the many diseases and parasites of the honeybee, it is the varroa mite which nowadays puts the greatest burden on beekeeping. This parasite damages the brood as well as the adult bees. In order to select for more resistant varroa-tolerant bees, the development of varroa populations is assessed and compared in testing stations. This investigation starts at the beginning of December by inoculating all stocks with an equal number of parasites. In preparation, heavily infected colonies are dissolved and shaken into swarm boxes. A sample of bees is removed and is later weighed in the laboratory, where it is also checked for infestation. The bees are washed in a soap solution, and this is passed through several sieves which separate the bees from the much smaller parasites. For easier counting, the bees are shaken onto a light-colored surface. The result and the known weight of the sample give us the absolute degree of infestation of the shaken swarm. The bees are fed with sugar syrup overnight. This ensures their acceptance when they are later added to the test colonies. Renewed weighing of the shaken bees allows a recalculation of the relative infestation and of the size of the bee sample needed to inoculate each colony. Several heavily infested stocks must be mixed when many colonies take part in such a survey, because each stock must receive a significant and equal number of parasites. The shook swarms are united and are thoroughly mixed. A large honey strainer gives sufficient ventilation for all. The individual dose can be calculated after pooling the various results of each batch.